Welcome to our first episode of Know Your Supplier. Today, we're going to introduce you to Celestial BioSystems and how Celestial BioSystems relate or collaborate with Inglo Biotech. Forgive me. So, Celestial BioSystems is the authorized agent for Inglo Biotech in the country, working in the capacity of its subsidiary in Zimbabwe. So, without wasting much time, we'll go straight into the business of Celestial Biosystems, and perhaps maybe before we go there, give you an understanding of who we are. First is Inglo Biotech, and then second is Celestial Biosystems. So first, Inglo Biotech is the African Genomics company. We have got our headquarters in South Africa and subsidiaries in West Africa and East Africa, and we have got representation in almost every country in Sub-Saharan Africa. So Inglo Biotech is a genomics company. We offer services. In DNA sequencing. So we actually do everything DNA. That means we are one, if not the most successful and the largest biotech company in sub-Saharan Africa. So I will start by mentioning the two arms of Innovative Biotech. So Innovative Biotech has um, services on the right hand side, and has got solutions in genomics on the left hand side. Um, to start with the services, so at Ingloba Biotech, at our headquarters in South Africa, we offer various services from DNA synthesis. As most of you would know, that we were the first company to actually produce primers, and like most of you would know Eppendorf because of their APs. You would know us because of the primers. So we are the primers company. We were the first to actually make and produce the primers. Um, so that is one of the product that put us on the map from 2002, and from then till now we have advanced our services. We had then Sanger sequencing, and now we have Sanger sequencing using some of the latest technologies. We have fragment analysis, where we do mostly genotyping, DNA profiling. That's where also our zoomics department comes from, where we do animal DNA profiling for the purpose of parentage testing. Um, uh, and many others. I will actually get into detail on on our services in the next few slides, because if I preempt it now, I may have to repeat the same things in the next few slides. So those are our services. One of our biggest services also is next generation sequencing, and we are the distributors for PEG Biosystems. So Sanger sequencing, next generation sequencing are the strongholds of our services departments. Um, the other arm is solutions. We are the distributors and suppliers of major brands. I would say housekeeping. Uh, should I say housekeeping or stronghold brands in biotech? So we are talking about Eppendorf. We are talking about New England Biolabs. We are talking about Zymo Research. We're talking about Glantham Life Sciences. We're talking about uh, Seegin, and many more, as I would explain and demonstrate in the next few slides also. So, as you can see there in the image that I just put up there, when you're in Zimbabwe, naturally in Zimbabwe, you would see in our biotech as Celestial Biosystems. I would take the glasses at the border. When you cross the border and go to South Africa, when you put on those same glasses, you will see uh, in our biotech. So that's, in summary, that's the relationship we have, and that's the image we depict in Zimbabwe, and that's who we are. So The next slide is on our mission. What is our mission as Celestial BioSystems in the country? So it's to become the leading life sciences and molecular diagnostics provider in Zimbabwe and beyond. We want to achieve these by granting all our partners, all our clients, all our uh, you know partners, access and success to molecular research. I think that way it is one of the that way it is one of the paths for us to realize our potential when it comes to advances in biotechnology in the country. So that is our mission and our vision as Celestial Biosystems. So now I'll also get back into the services that we provide as Ingloba Biotech. So Ingloba Biotech um, offers quite a number of services. I'll start with oligo synthesis, because that's where most of the DNA work starts. You definitely want to start with the primer. You definitely want to start with some amplification of your DNA. So in our biotech, we synthesize and make these primers. We do also assist in project for designing, optimizing primers, panels, um, and from there, one would either go the route of Sanger sequencing, the route of next generation sequencing, or the route of qPCR. So Sanger sequencing, as you'd know, is the gold standard of DNA sequencing. Um, we do offer Sanger sequencing services, um, and we also offer next generation sequencing using our PacBio systems, and we are also the distributor, as I said, for PacBio systems in sub-Saharan Africa. And I'm sure you would know that if you're dealing with next generation sequencing data, you would definitely want to utilize um, bioinformatics solutions. So we're talking about the bioinformatics softwares for data analysis. Um, so we do also, supply, we do offer, we do train um, scientists, if I may say, on bioinformatics, and we do also offer that and supply that with our next generation sequencing solutions. And we have got animal genetics, and I would want to combine animal genetics and fragment analysis. So maybe let me start with fragment analysis. So at fragment analysis, this is a service just like Sanger, but here we're not sequencing per nucleotide. Here we are sequencing per fragment size. So the fragment size of your DNA will give you, alleles, and then the alleles will tell you um, a profile, will give you information into the profile of your samples. So when you are doing animal genetics, we are using the same concept of fragment analysis, whereby we are running and analyzing fragment DNA of our target sample, of our target regions within a sample. So that's fragment analysis. And qPCR. Here we are dealing with real-time quantification of DNA, where they employ um, a reverse transcription or just ordinary qPCR. We do offer that, and also I would also demonstrate in the coming few slides that for all the products that we supply, we also do instrument services, and that is actually one of our strongholds, whereby we don't want to just supply an instrument to you and just leave it there to become a white elephant in your lab, but we do make sure that your instrument continues to function. So we do instrument services for all these instruments, all the platforms that we supply place at your laboratories. And the last one, even though listed first, is mass array. So here we are dealing with SNP genotyping, um, and various mass array solutions. I mean, information within each of these services is quite vast. So it is also helpful for you to get in touch with us so that we can look at all these individuals in detail. So we will proceed to the other arm of Innovative Biotech, um, which is solutions. So here we have got products for molecular biology, we have got instrument. We also now offer proteomic services. We have got plastic consumables, we have got biochemicals. We do offer quite a number of solutions on cell biology and my favorite, molecular diagnostics, softwares for bioinformatics analysis and food testing. Um, Next slide, we're going to run down all our, our solutions. And what I like to do here is to demonstrate using a workflow. So, um, what is a typical workflow for a scientist in the lab, a molecular biologist? Um, I would want to start from sample collection. And by the way, on our life sciences solutions and consumables, what you see there are the brands that we distribute, and we are sole distributors of in sub-Saharan Africa. So I'll start with sample collection. So we are the distributors for Zymo Research. So from Zymo Research, we have got sample collection solutions. Um, and around most, if not all, our sample collection solutions is um, the DNA RNA shield, which is a stabilizer that stabilizes your DNA at an RNA at room temperature and for transportation at that condition and storage at that condition for quite a considerable uh, long period of time. So around the swabs, around the the many solutions that you would use for sample collection, we have got at the center stage the DNA and RNA shield that functions to stabilize your nucleic acids before you can go on and do your nucleic acid extraction. So that is what we have on sample collection, and the solution that we have actually vary from different sample types. So the first sample type, I would say biological solutions. So we have got the swabs, and you would definitely, collect biological samples, not just with the swabs, but using different mechanisms and solutions. And second, we have tissues, we have um, plant materials, we have environmental samples, we have um, a typical one, which also falls under biological samples, are you know, the typical infectious diseases samples where we have got the swabs working the most. So on all those different sample types, we have got solutions from Zymo Research that you would use for that kind of sample collection. And then you from sample collection, you would definitely go straight into nucleic acid extraction. So our nucleic acids are our DNA and RNA. So let's say you're dealing with bacteria, you're dealing with fungi, you're dealing with viruses, you're dealing with protozoa, you're dealing with um, uh, insects, you're dealing with mammalian tissue, biological samples. We are dealing with pathogens. We have solutions that are optimized to extract nucleic acids from all those different sample types. So that's Zymo research for you, where you get pretty much every nucleic acid extraction solution that you would need. And I'm sure if your project, if your study would benefit from high yield DNA, RNA, high quality DNA, RNA, Zymo Research is the best brand to go to when it comes to nucleic acid extraction. I mean, I have used a number of kits for nucleic acid extraction, but nothing comes close to Zymo Research. So, uh, and by the way, if you want to try Zymo Research, we have got kits that we would send to you for trial. If you've got a project that would use much of our Zymo Research kit, we can offer you a kit, a sample kit to try and see for yourself how Zymo Research nucleic acid extraction goes and how the yield looks compared to maybe some of the different brands that you may have used in the past or that you're using now. So from your nucleic acid extraction, you would definitely want to proceed to do PCR. But before you do PCR, I'm sure you would want to check what is your quality of your DNA, what is the concentration of your DNA or your RNA. You don't want to be overloading that PCR. You don't want to also be injecting low quality, low yield DNA, RNA into your PCR. So you want to check that. So we do have got, and we are the suppliers of instruments, um, uh, from Thermo. So those are the nanodrops. So you want to do a nanodrop QC and check the quality of your DNA and your RNA. So if you're looking for a nanodrop, if you're looking for that kind of a solution, you can get in touch with us, and then we can do a product review, a quick online demo on on the nanodrop, and then from nanodrop QC you go to PCR. And by the way, um, complementary or supplementary solutions that you would want for nucleic acid extraction, whether in full or as kind of accessories, we also have that. Like, for example, you would have a kit maybe for tissue DNA extraction, but let's say, for example, you're dealing with bone tissue. We've got bashing beads. We have got, all those auxiliary or, accessory um, stuff that you want to use for nucleic acid extraction. All of those are still from Zymo Research. Then after nanodrop QC, of course, you'd continue, you go straight to PCR. So New England Biolabs is our brand for PCR, and it comes in three forms, if I may subdivide it like that. So we've got conventional PCR, we have got qPCR, and we have got next generation sequencing. We have got our NEB Next solutions for library prep, assemblies, um, and then we have got qPCR, we have got the Luna Universal qPCR, we have got the Luna Pro-based qPCR, we have got the Luna RT qPCR assays, and then on conventional we have got the famous one tag, two times master mix. I have used that one in the lab. I have used quite a number of Luna assays in the lab. It is quite interesting actually to see how the one tag amplifies across quite a number of different sample types. I mean, different DNAs. I mean, from the difficult DNAs with the high GC contents, DNA with long repeat motifs, all that kind of DNA that is totally different. So yeah, I can guarantee you that with the one tag, with the Q5, you never go wrong. And definitely after your PCR, you would either want to go and do your gel QC before you proceed with Sanger sequencing. You do gel QC, that's for conventional PCR. So we do have got solutions for gel electrophoresis from Thistle, um, uh, So that is you know, your gel electrophoresis, what do you call dams, your gel electrophoresis um, systems, uh, all those we do supply from Thistle, which used to be Clever Life Scientific. And we, let's say you do go on to do qPCR, I'm sure you would understand how to do qPCR data analysis. And then if it's next generation, then we are talking about uh, next generation sequencing data analysis. But within and between these, workflows, from sample collection, nucleic acid extraction, nanodrop QC, PCR, and then Sanger sequencing, you have got consumables that you use. You have got tips, you have got different plastics that you use. I won't delve into instrument because we're going to deal with that later on. So we do have uh, plastic that are used for these workflows across these different brands. You've got plastics, um, of course, from Zymo Research themselves, because those you would want to use them for nucleic acid extractions. But you've got tips, tubes, plates, strip tubes. Those are from Eppendorf and Treff. So that's where we get our plastics. And let's say, for example, you've got your own genetic analyzer in the lab. So you would, let's say it's an ABI 3500. You've got an ABI 3730. You've got the old, the new, and the latest ABIs. So we have got solutions for that from Nimagen. So Nimagen is a direct alternative for applied biosystem solutions. So yeah, and you have got Glantham Life Sciences, where we have you know chemicals. So this is the brand whereby when you're looking for any chemical, you'd find it from Glantham Life Sciences. So we're talking about, you know, the specialized chemicals. We're talking about, the housekeeping chemicals. We're talking about, you know, the routine, non-routine chemicals, specialized, non-specialized chemicals. All of these are from Glantham Life Sciences. And then Bioconcept, we're dealing with cell culture. You have got Rita here. We were dealing with liquid handling consumables for um, automation. And then we have got Euphoria Biotech, we have got Science Cell, we have got Biomatic, um, and many other solutions there. But also of interest is Charlotte. So here is where you get your microbiology solutions. So let me go straight into instruments. So instruments, these are instruments that we would use across the same workflows. So we've got Eppendorf as the first brand. So this is where you have your pipettes. This is where you get your centrifuges. This is where you get them your thermocyclers for conventional PCR, and as the distributor and the service provider for PECBio, we would definitely give you a service, one of the best. I think PECBio now, looking at other platforms, so next generation sequencing um, produces one of the best data that you would get after you do your next generation sequencing. So we do supply PECBio and we do offer services in PECBio. As I said, demo, that's where you get your nano drop, and you have got IKA with pretty much the most of accessory instrument, main instruments, of course, for a laboratory. And you could Vilba, where you do gel documentation after your gel QC, and you have got a faster. That's where we have got our laminar flows. So we have got cabinets. So that's for your microbiology, for your chemistry, and for the routine molecular biology. And then we have got glycochemicals research and Thistle. I'll combine these. Uh, they have got most solutions for gel electrophoresis. And then we've got cryogite for cryogenic storage of your samples. I'm sure you want to store your samples at -80. You want to store your samples for a very long period of time. And then we have got DNA star for your bioinformatics softwares. We have got Advita, we have got NanoLive, we have got Preciser for your um uh, balances, your moisture analyzers, and they've got BMS, which is biomolecular system. So from BMS, you do get your qPCR, the MIC, and qPCR platform, which is the MIC, actually, which is it is one of the latest technologies in qPCR that uses very interesting technology for fast cycling and for versatility in the lab and in the field. And so that's your qPCR platform. And we have got the Maya, which is the liquid handler. So we will in the next coming episode look at these individually, each brand individually, and perhaps maybe each workflow individual, and we'll definitely touch more on all these brands and what they have to offer. And we have got molecular diagnostics. So molecular diagnostics is one of our key portfolios, because from molecular diagnostics, that's where we also offer solutions that you know uh, that change lives in real time. So with the best if the best brand, the biggest brand is Seegin. So Seegin is our largest brand when it comes to molecular diagnostics. So Seegin offers you know highly sophisticated but simple uh, multiplex qPCR assays for 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 syndromic diagnostics. And it is here where you get your solutions for for diagnostics, surveillance, patient management across a number of syndromic pathogens, syndromic. That's where you get your syndromic assays. So here you have uh, HPVs, you have respiratory viruses, you have got STIs, you have got MTBs, you have gastrointestinal assays, you have drug resistant assays. So yeah, this is where you get the most of your diagnostic solutions. Uh, if you're dealing with clinical samples, human samples, and then we have got Divisor, for for some of the specialized diagnostic work, like prenatal testing, when you're trying to check the genetic condition of a preborn baby, yeah, some reproductive assays. We've got oncology. We are checking you know oncology markers. That's from Divisor. Then we have got Vercel. Vercel also have got the infectious disease assays like Seegin, and that's for clinical work when you're dealing with human samples. And you've got Mosam, so also in the same category, and they've got plant. So when you're dealing with plant solutions, plant diagnostics, plant diseases, yeah, you now look at BioRiver, and Seegin goes hand in hand with Biored. We use for most of our diagnostic solutions, we use Biored, CFX96, and very soon we'll be using the Opus. So it is also here where you also get your clinical diagnostic products too. And then we have been talking about infectious disease diagnostics for humans, but we also have got the same solutions for quite a number of infectious diseases in animals. I'll use a typical example, FMD. So we get all those solutions. So we're talking about Babesia, we're talking about Trichomonas, we're talking about Salmonella, we're talking about all these infectious diseases that affect our animals. Those solutions, you get them from gold standard diagnostics. And we have also got Ceramics. So Ceramics has got our panels for next generation sequencing. So we have got most of the oncology panels here also from Ceramics, and they've got Wayner for clinical chemistry, and uh, we have got clinical chemistry hematology solutions from Wayner Lab. Um, so I would want to perhaps stop there. Here is just a depiction of the high multiplex technologies that we have from Seegin across different syndromic assays. And this device. So I would want to stop there so that we can get time to look at this information and see how we can make it useful. So why choose Celestial BioSystems in Zimbabwe? We are very quick and efficient, and offer efficient services. We have got friendly staff. I'm sure those that have dealt with me, can attest to that. We have got local support. We are always at the client site. We are always there to support the client. We're always there to, listen and plan together with the client for current and future projects and see what solutions we can have. We definitely offer competitive prices. That's a given, and we are a one-stop shop for all your molecular work, for all your work with DNA. And in a partnership with Innova Biotech, we are definitely the leading, service provider solutions or chain supplier in biotech. So with that, thank you very much.